Hello, and welcome back to the lecture on Chapter 10. This will be part 6. Is that right? So part 6 of the lecture on Chapter 10. We will be looking at, we finished off the different mechanisms of action of the antimicrobial drugs, and we covered some of the problems. We talked about how broad spectrum the different drugs were. And then here were some of the problems with allergies and disruption of normal microbiota and why you would want a more targeted antimicrobial drug. In this next part, we will talk about the big problem that arises with antimicrobial drugs, and that is resistance. And as I've mentioned before, this is not a surprise. I mean, people always act like, oh my gosh, these bacteria are so smart and they're so evil and they develop these resistance. This is evolution. I mean, this is what happens. There's competition, and then there's, things that's the evolutionary arms race. One species does this to fight off another species, and then this species develops some, variations just naturally, spontaneously, independent of what this species is doing. But because this species is going to kill off anybody who doesn't any of this species that doesn't have the defense, then these guys die off, and then the one that has the variation survives and reproduces and leaves more copies in the next generation. That's how evolution works. And so this is not a surprise. So don't be surprised when all the antibiotics that we're talking about today, 20 years from now, there's resistance to every single last one of them, because that is how this is how the system works. Okay. So some pathogens, of course, are naturally resistant. They just have some ability to elude the drug. And then resistance can show up in a bacterial species in a couple of ways. One is, of course, by new mutations. Mistakes happen when the DNA is getting copied, and most of those mistakes get edited out and fixed. But some of the mutations stay for good, and usually they're damaging to the organism. But every now and then you get one that actually makes that individual better able to survive. And these mutations, if we're talking about bacteria, for example, they're going to be either on the main chromosome or they can occur on a plasmid, those little rings of DNA. And so let me back up for a second. So this stuff, if the mutation's on the chromosome or on the plasmid, that's going to be transmitted to the offspring, like the cells that divide, vertical transmission of this information. But remember, bacteria also have that trick where they can exchange plasmids with individuals in the same generation. So you can get horizontal gene transfer. And so you can get acquisition of these reproduction plasmids. That can happen via transformation, where the bacteria just pick up some like broken DNA, or transduction, where it gets actually transmitted by a virus that's affecting the bacteria, or it can be transmitted directly from one bacterium to another through conjugation, where the little tube, the pili, reach out. So first, let's cover vertical transfer of information. So here in this Petri dish, what we can see is like we inoculated the dish, and there is a drug that's in the agar. And what you can see is some of these cells are sensitive to this drug, and so they're not going to survive. They're shown here in this very pale color. If there's enough genetic variation in that population that just by happenstance, you happen to have some individuals who have resistance, natural resistance to this drug, what you've just done is created an environment in which anybody who's this darker color and has the ability to survive in the presence of the drug, well, woohoo, now the competition's been taken out, and now this plate is ours, and we will grow and grow, and this, everybody else is dead, and there's this one that's kind of limping along, but these populations are really going to take over. And so the sensitive cells get inhibited by the exposure to the drug, and now you've got giant populations of resistant cells. This is what happens in hospitals and how we end up with so many nosocomial infections that are resistant to drugs, because the drugs are being, like the patients are being exposed to the drugs. And so all of the bacteria that are growing in those patients, the ones that are sensitive to that drug, die off. And it's only the resistant strains that survive. And then that's what gets kind of smeared onto the handrail or onto the light switch or onto the whatever. And then it gets passed inadvertently, it gets passed from one patient to another. And then we have a patient who came in for a hip replacement surgery, but is exiting with pneumonia, or ends up even sicker because now they have pneumonia that is drug resistant. Okay, so that's a vertical transfer of the mutation. And then this is the explanation for why patients are strongly urged every time to complete the course of treatment when they're prescribed antibiotics, because you're trying to avoid the situation where you killed off some of the microbes, but not everybody. Because it's possible, like if you had a different drug that could kill this off, that could kill these off if we took the medications for long enough, we would be able to kill off at least this guy. I'm not explaining this very effectively, but essentially you're creating a situation where you're knocking out the competition and favoring those microbes that are resistant. And so what I'm talking about down here, complete the course of treatment, let's say they weren't this robust, that they were like this guy that could survive a little bit but wasn't doing so well. And then if they just kept exposing, for the full course of 10 days, that might eventually kill that guy off. And then you wouldn't have any bacteria that are resistant to this. Okay, and then this is an example of horizontal gene transfer. So once the mutation that leads to resistance occurs, it can be transferred horizontally. Here, the example that's being shown is a bacteriophage. And anyway, in other words, there's a vector that's going to take the resistance gene from this bacterium and insert it into another bacterium. And so the bacteria can acquire resistance that way. It could also be through transformation, where the bacteria just pick up kind of bits of DNA, or it could be through conjugation, where the two bacteria come together and exchange a plasmid, or plasmid passes from one bacterium to the other. And we have already identified that there are multiple mechanisms of resistance. I've already told you about one, and that's this one, the production of an enzyme that destroys or deactivates the drug. The drugs of interest would be beta-lactam, like penicillin or amoxicillin. And some bacteria have now evolved a mechanism, an enzyme that can actually just snip that beta-lactam ring. Those are the beta-lactamases. And right now we have a defense for that, and that is the clavulinate. It's the mechanism that it's the inhibitor of the beta-lactamase. So the little scissors, and we've got the rock against the little scissors. But there's some bacteria that have already evolved defenses against that. Again, not a huge surprise. Some bacteria have evolved mechanisms to slow or prevent the entry of a drug altogether. The target of the drug, the protein or whatever it is that our drug is targeting, if a mutation arises in a bacterium where that protein changes its shape a little bit but can still do its job, it eludes the drug that we're trying to kill it with, but can still do its job. And so that's another mechanism of antibiotic resistance or microbial drug resistance. Some of them actually develop pumps. I kid you not, they're transport mechanisms. They're, the bacteria pay for it with ATP, and it pumps the antimicrobial drug right out of the cell before the drug can actually have its effect. They're called efflux pumps. And finally, you remember biofilms, the bacteria can kind of create these colonies where they're physically laying on top of each other and protect each other physically. The outer layer of the biofilm is exposed to the drug, but the inner layer is protected. Okay, and there are other mechanisms. The point is that this is an evolutionary arms race, and it will always be an evolutionary arms race. We will never develop a drug that there won't be a way for bacteria to elude. There's always going to be variation coming up through mutation, and then if that variation benefits the bacterium, then it gets selected for. Okay, this diagram is just illustrating this point about the beta lactamases. So there's here's a beta lactam penicillin. There's that important beta lactam ring, and the beta lactamase, the enzyme that the microbe can have, snips that connection here between the carbon and the nitrogen. And now this ring no longer works. It does no longer interfere with the connection of the NAM units to each other. And so that bacterium can happily continue building its cell wall. Clever, clever. All right, now multiple resistance. I've mentioned that there's multiple ways of, we've already seen that bacteria have different ways of eluding these different drugs. And it's possible for a single pathogen to acquire resistance to more than one drug. We call it multiple drug resistant bacterium. This is super common where our plasmids, those reproduction plasmids, where those can be exchanged through conjugation. Oh my gosh, so all we have to do is have resistance develop in one strain of bacterium and a different form of resistant strain develop in another form of bacterium, and they can pass that information to each other even across species. Crazy making. This is especially common in places where there's going to be a lot of antibiotic use, places like hospitals, places like, dairy farms, places where a lot of animals are crowded into a small space, and there's going to be lots of infection, and there's going to be lots of use of antibiotic. Again, it's not the antibiotic that causes the mutation, it's the antibiotic that is wiping out those microbes that are sensitive to those antibiotic and allow selection for the ones that are resistant, like we saw in the Petri dish with the big red colonies growing. So hospitals, nursing home, any kind of rehabilitation facility where people are, packed together for a long time. And so again, the constant use of these antimicrobial drugs eliminates the sensitive cells. That's what's supposed to happen. Unfortunately, it creates the perfect conditions for any cell that happens to have some resistance. Wow, those can flourish. When we say that a microbe is multi-drug resistant, we're talking about resistant to at least 3 different antimicrobial medications. Now, to retard the development of resistance, to slow down the development of resistance, we know it's going to happen, but we know what conditions select for it, and that is kind of chronic low concentration use of antibiotics, which seems like a good idea, but it's terrible in the long run because it wipes out the sensitive cells and selects for these resistant strains. So what we do is we maintain high concentration of the drug in a patient for long enough period of time. We can use antimicrobial agents in combination. So maybe this microbe has a little bit of resistance to this drug and maybe a little bit of resistance to this drug, but when combined together, cannot fight those two antimicrobial agents together. This is referred to as synergism, and I have an image to show you. This is a Kirby-Bauer plate showing bacterial lawn. Here's one antibiotic. Here's a different antibiotic. They have the zones of inhibition, but can you see how big the zone of inhibition is in that field where both chemicals are present? Wow. This bacterium doesn't stand a chance against this combination of antimicrobial medications. Also, we use antimicrobials only when necessary. This has been the general movement over the last few decades. Healthcare providers are no longer prescribing antibiotics just for any old thing. It used to be anybody came in with a sniffle, automatically got a prescription for antibiotics. We do not do that anymore. It's inconvenient in some ways for the patient in that moment because they think, I'm sick, I want to try anything that might help me. But if you have a viral infection, you have a really bad cold, or you have the flu, giving you antibiotics isn't going to help you anyway. So in the long run, it is beneficial for patients to have the provider not be prescribing antibiotics for everybody, so that in the community, we don't select for resistance. In other words, we don't wipe out all the sensitive bacteria and just have the really hardcore bacteria that are not going to respond to drugs. Another technique is to develop new variations of existing drugs, second generation drugs. For example, the beta lactams and the other the adjuvant, the thing that you would add in addition to the first drug. We're up to third generation drugs. I mean, you have fourth generation drugs, and this will go on through your entire career. And finally, the search for new antibiotics, semi-synthetics, and fully synthetic antimicrobial agents. Bacteriocins, the idea here, the research in this area is going into bacteria themselves and saying, when you are in your natural habitat, how do you fight off other bacteria? We want to know what what molecule you're making so that we can make that molecule and we can use it. And then we can do this in some ways more sophisticated, but in other ways less effective and a lot more expensive approach, which is to actually design drugs. And the way we do it is look at the shape of the microbial proteins. Let's see what the machinery is that the microbe needs to do its jobs to survive and reproduce, and then build molecules that are shaped in a way that's going to interfere with that protein doing its job. Yeah, so it's kind of like doing ourselves artificially what nature has been doing for millennia with the bacteriocins. All right, and that is the end of this part. It says part 5, but it should be part 6. That's the end of the information from chapter 10. Hope you have a great week. I will stop recording in a moment. All right, have a great week.